Good morning, ladies and gentlemen. The topic I am going to talk today is HLA system. Stands for Human Leukocyte Antigen System, which is the histocompatibility antigen system of mammalian cells. Now, the pathway to the discovery of HLA started by surgeons actually. Now, this was in the context of organ transplantation. Organ transplantation has been practiced by surgeons centuries ago. Started maybe more than 3,000 years. The description of transplantations are there even in our ancient Indian medical literature. But most of these transplantations fail. People didn't know the reason why they fail. Now, in early 20th century, Alexis Carrel, after a series of failures of transplants, Carrel did some vascular corrections and tried some kinds of transplantations and was successful in some of them, and he won the Nobel Prize for his work. In 1906, corneal transplants were materialized, and then it became a practice without much of a failure. But subsequently, most of these grafts failed. That was a series of failures, and that led to the drop of organ transplants. People didn't know, but surgeons continued trying transplants because many of them thought grafts between two trees, say fig trees and olive trees, there was no rejection by the trees, and human beings are not that different as the trees. So they continued trying, but the failure rate increased. So gradually the attempt at transplants came to a stop, except for a liquid graft, namely blood transfusion. All of the grafts, they almost were discontinued. So only some of them succeeded. The way they only the blood transfusions remained, and most of the other transfusions came to gradual decline. But later on, during the Second World War, the need for transplants, especially skin grafting, was increasing tremendously. And at that time, one of the British zoologists, namely Peter Medawar, he studied these grafts which were failing and the grafts which were succeeding, and he postulated that the rejection was mainly by an immune mechanism. People till that time didn't realize the importance of lymphocytes. Lymphocytes were a bland-looking cells, and they didn't find much importance of lymphocytes. People knew about neutrophils and macrophages, but not about lymphocytes. So seeing lymphocytes was not very capturing for the biologists at that time. But Peter Medawar worked on this and postulated the immune theory of graft rejection, and he postulated all the works or the mechanisms of graft survival, and received Nobel Prize in his work. Right. Now, what are these antigens? Human leukocyte antigens. They are actually antigen recognizing molecules situated on cells. Most of our cells do have on the surface these antigens, because cells can't speak, cells can't hear, cells can't see, but they communicate with chemicals. But there are molecules which make them communicate between each other. So cells recognize between them, between each other, by the use of certain molecules, and HLA molecules are among them. They are antigen recognition molecules, or in a way they are the identity cards of cells. Now for the cells, for their identity, they have two choices. They can have a class 1 antigens or a class 2 antigens. MHC stands for major histocompatibility complex, which is the genetic locus for the coding of HLA antigens. We will come to that later. So either class 1 MHC or class 2 MHC molecules are there. Class 1 MHC molecules are virtually present on almost all cells except the red cells, red blood cells. Including platelets. So important that for platelet transmission also, that is a critical thing. Whereas MHC class 2 has a limited distribution. Class 2 molecules are expressed on certain cells which are involved in antigen presentation, like the macrophages, monocytes, the dendritic cells, the Langerhans cells, and the B cells, B lymphocytes. They are the cells on which MHC2 molecules are present. So all the cells do have class 1, but these antigen presentation involved, all cells involved in antigen presentation have both class 1 and class 2 molecules. Now the function of these two molecules, class 1 and class 2 molecules, differ in that the class 1 molecules present antigens processed within the cell upon their surface for presentation to the cytotoxic T cells. Peptides processed within the cell means cytosolic proteins which are there within the cell, or proteins which are derived from external agents, but the cells have taken them up, they are processed inside. Those antigens are expressed on the surface for presentation to cytotoxic T cells, and the T cells can recognize them and deal with these cells. And this usually calls for a destructive signal, because it is dealt with cytotoxic T cells, cytotoxic cells. So they are cytotoxic, and therefore cells containing such, or cells expressing those unwanted antigens have to be deleted then and there. They have to be eliminated. So that is the function of the cytotoxic cells. So presenting such antigens, such cytosonically derived antigens, calls for a destructive signal. So that signal is taken up by the cytotoxic cells, and this kind of cells are therefore eliminated by the cytotoxic T cells. That is the reason why all nucleated cells have class 1 MHC, because for example, any cells can be infected with viruses. There are no exceptions. Any cell can be infected by viruses, and virus infected cells process those viral antigens within the cell and express them outside. And there are viral antigens are there for exogenous antigens, but there can be endogenous antigens, like the cells get changed by, say, some kind of internal problems, like cancers. Cancers cause a modification of the cellular environment, the cellular structure, and abnormal antigens are expressed on the surface. And these are all also regarded by the immune system as non-self. Actually, immune cells have to recognize self and non-self. So whatever is not belonging to us are regarded as non-self. So elimination of such non-self is a function of our immunocompetent or immune system, our defensive system. So those carrying viral antigens, those carrying cancer-related antigens are all considered as unwanted or non-self material. So cells carrying such unwanted or non-self things have to be eliminated. That's why all the cells are equipped with this class 1 MSC molecules, because cytotoxic T cells can deal with any of these cells. Whereas the class 2 molecules expressed on a select population of cells, the antigen presenting cells and the B cells, they are derived from peptides digested in the endosomes, and they are expressed on the class 2 MSC molecules. And class 2 MSC molecules, they present the T helper cells that can only be read by the T helper cells, not the cytotoxic cells. Molecules expressed on class 2 molecules class 2 MSC molecules can be identified, can be read by only the T helper cells. And that calls for a help signal. The T helper cells, recognizing them, they can process it, they can send the messages across the macrophages to produce cytokines, or they can direct to the B cells to produce antibodies against this. So that is a function of T helper cells. So these are T helper cells are CD4 cells. Cytotoxic cells are CD8 cells. So class 1 is a CD8 restricted and class 2 is a CD4 restricted molecule. So this is the genetic map of MSC locus. It is situated in chromosome 6, a small locus but very tightly packed cluster of genes in chromosome 6. So it has class 1 region. Class 1 codes for 3 separate genetic loci or genetic regions A, B and C. So class 1 molecules include A, B and C. Class 2 molecules include the D region, which is subdivided into DP, DQ and DR. And there is a class 3 which are not actually HLA molecules. They produce various cytokines, complement proteins, etc. They are not directly involved in antigen recognition, but they also form part of the machinery for dealing with these peptides. But actually antigen recognition is done by these two, class 1 molecules and class 2 molecules. Class 1 ABC, class 2 DPDQDR. And these are extremely polymorphic genes. Why? Because you just see that A, B, C, A, this is the statistics from 2016 or so. HLA-A, 3,657 different types of HLA-A alone. 4,459 HLA-B alone and 3,290 HLA-C alone. See, there are many more to be still recognized. So you can just calculate this many number of HLA MSC1 molecules are available. And you just see one individual inherits one allele from the father and one allele from the mother. One HLA from father, one HLA from mother, one HLA-A from father, one HLA-A from mother. So there will be 6 MSC1 molecules in one individual. Similarly, HLA DP, DQ, DR also there will be another 6. So altogether, all these will constitute a very wide variety of HLA molecules. Means the combination in one will be totally different from the combination in another. And two similar HLA combinations occurring in a random at random in any individual is so remote, means it will be very rare to have a suitable or a similar combination, an identical combination. So one HLA combination, HLA set or HLA haplotype we call, one person having the combination of HLA molecules in one individual is a haplotype. So one HLA haplotype in one individual is totally different from the HLA haplotype in another. That is the reason why the rejection happens. There is total mismatch of HLA between individuals of even the same species. And this is the structure of those HLA molecules. Why is this kind of the polymorphism? How is the polymorphism possible? This is the HLA class 1 MSC1 molecule. MSC is the gene locus and HLA is the product. The MSC or HLA class 1 molecule appears like this. It has an alpha chain and a beta chain. The alpha chain is the HLA product. Beta chain actually not a HLA product, it just is an associated molecule, but it is derived from a different chromosome, not from MSC, not from the MSC gene at all. But it is needed there, it is to support and to anchor it to the cell membrane. So there is an alpha domain, an alpha 1 domain, alpha 2 domain, alpha 3 domain, 3 alpha domains are there. Of this alpha 1, alpha 2 interface is the place holding the antigen. Class 2 has alpha and beta chains, both derived from MHC, and this is the alpha 1, there are alpha 1 and alpha 2 domains, beta 1 and beta 2 domains, and antigen is received in the interface between alpha and beta. Okay, so that is the difference, but how is the polymorphism possible? See, alpha chain, beta chain, they are all peptide chains and all composed of a series or a sequence of amino acids. The amino acid composition here is, say if this is, you are considering HLA A1. A1 will have a set of amino acids here. A2 will have a set of amino acids which are different. A2 amino acid constitution will be different. A3 another still different. A4 still different. Like that, all these more than 3000 types of A will have different constituent amino acids. That means the way they bind an antigen will be dependent on the amino acid constitution. Okay, so that makes it different, that makes it polymorphous. One HLA A is different from another HLA A because of this difference in the amino acid constitution. That means they can bind different antigenic peptides or different antigenic sites. So that makes it very much diverse. The same way HLA MSC2 also, the same kind of diversity is applicable here. Just little more about class 1 MHC. There are hundreds of alleles. Alleles means the genetic variants for each class 1 gene. I just explained class A1 is different from A2, A2 is different from A3 by the constitution of the amino acids. We have the maternal and paternal alleles in an individual, and the likelihood of finding two randomly selected people with identical HLA is 1 in 70 billion. You know what is the world's population? Right now it is about 8 million or so, 8 billion. So it is virtually impossible to find a HLA identical, random HLA identical person. Of course, identical twins will have the similar HLA. Other than that, if you randomly try to select an individual from the population, you won't easily find one. Identical twins have an exact match. Now class 2 MHC, we saw 2 is a heterodimer, an alpha chain, a beta chain, and this is a peptide binding groove. I told you there is a limited distribution. Distribution is only on the antigen presenting kind of cells. They are little more complex proteins. And the alpha and beta chains are encoded by different genes in the same locus but different genes. And a perfect matching by random selection, just like the MHC1, is close to 0. It is very hard to find an identical match. Now, why this polymorphism? Why should I have HLA so much polymorphic? Now, we see that HLA molecules are not only present in human beings, it has been conserved from time immemorial from the lower class of animals, lower class of living beings. It has been carried through evolution. Even advanced forms like human beings also have same HLA molecules. And what does it mean by their conservation over these billions of years? The conservation through evolution means there is a survival advantage, otherwise there is no need. Why HLA molecules become so complex to confuse students, doctors and all? It is because there is a conservation advantage over time among the evolving species. And the range of peptide binding is extensive. See, the antigenic diversity makes it possible to bind any kind of peptide molecules there. If one individual is not able to bind one particular antigenic peptide, another individual with a different constitution may be able to bind it. That means it is possible to say if you are talking about a bacterial peptide, that bacterial peptide may be bound by one of these individuals. If not one, it may be done by another. That means the reaction is made possible by some individuals, and that individual's progeny can have a little more bigger armamentarium against such an antigen, and that makes the immunity to that organism possible over a few generations. So that is a big advantage. So that is the reason why the HLA remains evolving over time and becomes more and more complex, more and more diverse. Each of us having different class 1 and class 2 molecules means different reactivity to the same antigen. The different constitution of each MHC molecule makes the reactivity different. So one individual reacts to one antigen a way different from another individual. For example, if a pollen particle or an epitope of a pollen grain is the one causing a reaction in one individual, he may react positively with a hypersensitivity reaction. The same pollen may not evoke any response in another individual, and that person is silent or that's not reactive to that because it doesn't recognize this peptide molecule or the appropriate molecule on the pollen. So that reactivity is different. Some may have a minor reaction, some may have a major reaction. So that accounts for the different kind of reactions to certain drugs. For example, one person developing an anaphylactic reaction to a penicillin, another person, majority of the people may not react at all. Some people develop a minor reaction. The difference is because of this diversity. If not polymorphic, what can happen? A cunning parasite with an identical HLA identical to the host can easily masquerade itself, can remain like a self. It can be accepted as a self, and it can multiply without any problem in that host and can even eradicate the human race. Theoretically it is possible. So just think of that possibility. So that is the reason why nature has evolved a method to make it more diverse and escape or defend the body against invasion of any such organism. So that's one of the reasons why it has become such complex and diverse. Now, HLA, what is its role in graft survival? Now we are living in an era of organ transplants. So many different types of organs are now being transplanted, apart from the routine kidney, bone marrow, skin, etc. We are now dealing with heart transplants, transplants of lung, liver, then pancreas, like that. Many organs are now being transplanted. One word, the dictum is that better the match, better the survival, no doubt, because HLA matching, just like blood group matching for blood group compatibility, HLA matching is the key word for the organ matching or the matching of hosts. The better the match, the better the survival. If there is a perfect match, there is nothing to worry. And of these HLAs, A, B and D are as the most essential. That those three are the most crucial ones deciding the matching, the survival of the graft. Matching is usually not done for all organs like heart, liver, etc. because there are other priorities for these organs. The anatomic compatibility, the time duration of holding the organ after removal, etc. They have more priority than looking for the match, because HLA matching is not done in one day. It takes time. And also because of immunosuppressive therapy now which is available nowadays, that takes over. That can cover up the shortage in the HLA mismatch to a certain extent. Lot of newer generation immunosuppressive drugs are available, started from steroids, then many other drugs, cyclosporine, the newer variety of calcineurin inhibitors are all available nowadays. So those are now taken care of in most of these cases, and therefore the graft survival has now increased to a great extent. Now there are certain diseases with HLA association, and most of these diseases you can see are inflammatory diseases or autoimmune diseases, like ankylosing spondylitis, hereditary hemochromatosis. These are a few among the list. There is a very strong association of B27, HLA B27 with ankylosing spondylitis. A person carrying B27 has 175 times higher chance of developing ankylosing spondylitis than person without this HLA. Like that, all these are different HLAs with somewhat strong associations with certain diseases. Now just understand why this association is because the inflammation causing molecules or the molecules responsible for inciting inflammation, they are continuously being produced by the body. That's why the inflammation is persisting over a long time, and those molecules are able to be detected by the HLA molecules, and the body can react against them. Just because autoantibodies are present doesn't mean that they cause the disease. They can be present even otherwise. Even normal individuals can have the autoantibodies. That doesn't necessarily mean the person can develop the autoimmune disease, but the presence of them persisting and capable of getting detected by the HLA and eliciting a reaction which can persist. that goes on to the particular inflammatory diseases. So, these are the common ones among them. There are many more, but it is not necessary that all of them are to be remembered. So, that is the essence of HLA system I wanted to convey. Thank you very much.